Well, good day, everybody. I'm Russ Reed, and I'm the Executive Director of the National Center for the Biotechnology Workforce, located here at Forsyth Technical Community College in Winston-Salem, North Carolina. And today we're going to have a lecture for our Biomedical Emerging Technology Applications grant from the NSF ATE division. And I'm delighted today because we have with us Dr. Tracy Criswell, who is an associate professor at the Wake Forest Institute for Regenerative Medicine, which is a part of Wake Forest University and is located right here in Winston-Salem and right in the research park where I am at, called the Innovation Quarter. Good day, Tracy. How are you?
I'm fine. How are you?
I'm very well, thanks. And at the helm for the media is Sean Killebrew, our master at doing this. And Tracy, I understand you're going to talk to us a little today about tissue engineering and regenerative medicine.
That's correct. I plan on giving a general overview of the field and talk about some of the applications and some of the technologies that are also being used that are coming out of the field.
Well, thank you, Tracy. We really appreciate you doing this with us, and I'm going to turn it over to you.
Okay. Thank you. All right. Well, thank you for inviting me to talk today. I am going to give an introduction to tissue engineering and regenerative medicine. I'm an associate professor at the Wake Forest Institute for Regenerative Medicine, and I've been there for over a decade now. And I really enjoy talking to people about what we do there. So I did list a couple of learning objectives for today, so that we can all be on the same page. But basically, I hope that you guys leave with a general understanding of what tissue engineering and regenerative medicine are. I'm going to discuss some of the barriers that we face to using TERM in the clinic. And then I'm going to end by talking about some of the enabling technologies that are coming out of tissue engineering that are being used in other fields as well. So the first thing that we have to do when we talk about tissue engineering and regenerative medicine is define the terms. So what is tissue engineering? Well, tissue engineering is an interdisciplinary and multidisciplinary field that aims at the development of biological substitutes that restore, maintain, or improve tissue function. I like to think of this as the outside the body approach. So we're engineering an organ or tissue to replace a diseased or dysfunctional organ. Regenerative medicine, on the other hand, is a broad overview. It's a broad field that includes tissue engineering, but it also incorporates research on self-healing, where the body uses its own systems, sometimes with the help of foreign biological materials, to rebuild tissues and organs. And this is more of an inside-the-body approach. So this is where stem cell transplantations would come into picture. And just so that we're all on the same page, just a brief overview. Cells are the building blocks of all living things, but single cells don't work alone. Cells work together in groups called tissues, and then tissues come together and work as organs. And so, for example, the heart is an organ that's made up of cardiac muscle tissue, and the tissue is made up of cardiac muscle cells. As we get older, as we age, our organs don't seem to work as well. There are degenerative diseases that affect the heart, the brain, joints, and skeleton, your skeleton. Injuries, so infections, trauma, environmental exposures. And then there are immune system attacks, cancer, things like this, that tend to accumulate as we get older and result in organ failure. And the problem with that is that organ transplantation is a zero-sum game. And what that means is that if you look at the graph at the bottom here, red are patients waiting for an organ, blue are the donors. And so you can see there's a massive gap, right? So there aren't enough donors for the people that need replacement organs. And that's where tissue and engineering and regenerative medicine could come into play. So I like to start with this guy, the newt. The newt is pretty cool. So if you cut off his leg, it will grow back to be completely identical to the leg that it lost in about 90 days. So the newt and a lot of salamanders have this incredible ability to regenerate their tissues. Now, people, this is a paper cut, and we know that a paper cut's going to heal pretty well. Our body can handle small injuries. However, it's these large injuries, like this volumetric muscle loss, that our body can't handle. And it's for these type of injuries that we need some alternate approaches. So that leads to how do we do this? How do we engineer an organ or a tissue? And there's really a trifecta of requirements. So one, we need a scaffold, we need cells, and then we need physical factors, chemical and physical factors that inform the cells of their physiological function. And I'm going to go into each of these components in a little more depth. But before I get to that, I want to highlight the complexity of organs and tissues. So tissues are made up of more than just cells. They have a vasculature, they have neurons, there's a tissue architecture that's required for proper function. There are mechanical forces that act on the tissues. There's shear stress through the blood vessels. And then the cells, the different types of cells talk to each other. So they don't live in a vacuum. And so when we talk about tissue engineering, all of these different aspects have to be taken into consideration. And that can be an overwhelming challenge at times. And so this, in an ideal world, this is how our tissue engineering paradigm would work. We'd have a patient. We'd take a biopsy from that patient. We'd isolate the cells and grow them up. We'd place them on a scaffold, let them mature into a tissue, and then transplant those back into the patient. So I'm going to start talking about scaffolds. This is the first component I'm going to talk about. So what is a scaffold? Well, if you look at this picture, this is a house. We know that this is a house, although it doesn't actually function as a house yet, right? It doesn't have walls. If you sleep in it, you're going to get wet, cold. Animals can walk in. There's no protection. But we know that this is a house, and that's what a scaffold does. A scaffold provides the support for the biological components, the cells, to grow on. And so there are different types of scaffolds that we use. Some are from synthetic sources, and these would be man-made sources, and then some are from natural sources. They come in various textures and materials, and the scaffold that you use is going to be dependent on the tissue or organ that you're trying to engineer. But all scaffolds have three requirements. One is that they need to allow nutrients to permeate. That means that the cells that are grown into that scaffold have to have oxygen and nutrients to survive. It has to encourage cells to grow. And of course, we don't want to use any sort of scaffold that would harm the patient. It's counterintuitive. And so if you look at these different scaffolds in this picture, you can see that they come in all different shapes and sizes. So if you want to engineer a bone, you're going to use a scaffold like this, which is hard and weight bearing. However, if you want to engineer the cornea of an eye, you don't want to use the bone scaffold. You're going to use a hydrogel like this that fits the function of the eye. Blood vessels, obviously, you're going to use a tube-like structure. And so, once again, the scaffolds that you use are dependent on the tissue that you're engineering. One method of engineering scaffolds is using electrospinning. Electrospinning is diagrammed here, where it's got a polymer that is released towards a conductive plate and can form a structure on that plate, whether it be tubes or a flat sheet. This is actually a diagram for a flat sheet, as you see over here. But the benefit of electrospinning scaffolds is that you can control the size of the pores in that scaffold and the size and alignment of those fibers. And why is that important? Well, if you look at this paper published in 2014, this is for skeletal muscle stem cells. And what they showed was that if they use a misaligned scaffold, electrospun scaffold, the cells grew like this. So the cells are misaligned. But if you use a scaffold that has these fibers that are aligned, then the cells align with the fibers. And this is very important, especially for tissues like skeletal muscle, where all the muscle fibers have to be aligned or you won't get proper function. Something that WFIRM has been involved in quite heavily lately is tissue decellularization. And so I've got a liver, a kidney on the right and the liver on the left. Basically, what this does is this takes a tissue, the diseased tissue, and then removes all the biological components. And it does this using detergents. We wash all the biological components away. You can see the liver, and then it becomes this ghost-like structure. But it maintains the scaffolding, the natural scaffolding of the liver. And you can see the blood vessels that are also maintained. And the green images below that are really a dye that had been injected into this liver scaffold. It shows the vasculature. We've also done the same thing with a kidney. We use porcine kidneys, pig kidneys, quite often for this because their kidneys in the pig are pretty much the same size as kidneys in the human. And what we've done is we decellularize it. We show that the vasculature is patent or is still there. And this has actually been put back. These are put back into pigs. And what we're showing here is that they're able to get blood to flow through the kidney after it's been recellularized. So the important points to take away from this is that scaffolds provide support and structure for cells and engineered tissues. They can be derived from natural and synthetic sources, and the material used is dependent on the tissue to be engineered. Whole organ decellularization provides a scaffold with preformed structure and vasculature. And I'm going to leave this with this question, which we'll answer later. Why is vascularization important? So if we were in person, I would ask you what this is. But since we're not, I will tell you that this is a 4-egg or 4-cell stage of a starfish, a echinoderm. So I'm a cell biologist by training, and I find this is what gets me up in the morning, is that from those 4 cells, and actually from 1 cell, all of the cells and tissues in our body develop. And we all start the same. Every animal on the planet starts as a single cell. And from that, all of our differences arise, all of our different tissues. How does the body do that? This is fascinating. So in case you hadn't noticed, we're moving on to cells. The official definition of a cell, I like to quote Dr. Watson, and it's the fundamental unit of life, the smallest body capable of independent reproduction, and cells are always surrounded by a membrane. And I like that last sentence, that cells are always surrounded by a membrane, because it seems really obvious, but it's one of those things that's so obvious we never think about it really. That membrane's important because if the membranes weren't there, it would just be essentially bags of water on the ground. So stem cells, what is a stem cell? Stem cells that we use in regenerative medicine. There are 3 properties to a stem cell. The first one is that it can replicate itself or repopulate the stem cell pool. So if you injure yourself and your stem cell gets activated, you want to keep those stem cells there for the next time you injure yourself. So it can replicate itself, while at the same time, it can make daughter cells that then differentiate into other cell types. And that's called asymmetric division. So it divides into 2 different types of cells. And then a stem cell also shows potency, meaning that it can differentiate into other cell types. Stem cell terminology. Stem cells can really be defined by two different aspects. The first one being their potency. And so totipotent stem cells really come from the zygote early embryonic stage. A totipotent stem cell can form the embryo as well as all the extra embryonic tissues such as the placenta. Whereas pluripotent stem cells, these are the embryonic stem cells that are commonly talked about. These come from a later stage embryo, and they can form all of the tissues in the body, but not the placenta, the extrauterine tissues. Multipotent stem cells, these are cells that are lineage restricted, meaning that they can either form all the cells and tissues from the ectoderm, or all the tissues from the endoderm, or all the tissues from the mesoderm. And then lastly are unipotent stem cells. And these are really what we use the most often. These are the adult stem cells. So if you injure your muscle, it's the skeletal muscle satellite cells, which are the stem cells, that become activated. And those are unipotent stem cells, meaning that they can really only become one type of cell. Where do these cells come from? Well, most tissues in your body have stem cells. They have adult stem cells that are waiting for injury. They're waiting to be activated. And when we talk about using stem cells for transplantation as a clinical therapy, there are really two issues that we take into consideration. One would be, are they autologous stem cells? So these would be cells from the patient being put back into the patient. Or are they allogeneic stem cells, which means they are human stem cells from a donor being put back into a human. The problem with allogeneic stem cells is that you will get rejection of the tissue. And so the patients that undergo this type of stem cell transplantation have to be on anti-rejection drugs for the remainder of their life. The last type of stem cell I'm going to briefly draw your attention to is the induced pluripotent stem cells. And I bring this up because you've probably heard of them. So this is a really interesting technology. They can take a cell, say a skin cell, and with 4 different genes, you see this down here, so fibroblast or skin cell. These are somatic cells, not stem cells. You can reprogram those cells using several different transcription factors. And what that basically does is it takes it back in time. It increases its potency now. They de-differentiate into pluripotent stem cells, which can then be programmed to differentiate into cardiomyocytes, so cardiac stem cells, or skeletal muscle stem cells, or neural stem cells. So the nice thing about the iPS cells is that you can take a patient's own skin, take some cells from the skin, grow them up, differentiate them into muscle, and then use them to treat a muscle defect they might have. And so this would be a source of autologous transplantation. Now the issue with iPS cells is that most of these transcription factors, these genes that we use to de-differentiate the somatic cells, are also tumorigenic. So they've been known to be involved in the initiation of cancer, and people are hesitant to use cells that have ever expressed these factors in case they may form tumors. And so until that's really cleared up, you're not going to see this you're not going to see this technology used in the clinic as often as it may be in the future. And so I want to leave here with the understanding that different types of stem cells perform different functions. So these are satellite cells that were taken from a rat. These are skeletal muscle progenitor cells. And these are from a rat, but they're same if you isolated them from human muscle. They know what they're supposed to do automatically. So these are cells that are growing on a plastic dish outside the muscle. And if you put them in the right condition, hopefully you can see where these arrows are pointing. They spontaneously contract like muscles are supposed to do. And so it would be a bad idea to take these skeletal muscle cells and try to use them to regenerate a liver, right? Likewise, we wouldn't use liver cells to try to engineer a heart. So the stem cells know the tissue they came from, and so when we engineer tissue, we have to be able to isolate the appropriate stem cells for the tissue we're trying to engineer. And I want to talk about Planaria. This little guy has become one of my favorite models of regeneration. He's a flatworm. He's a really simple type of creature. He grows to be about maybe an inch in length at the most. He's found in all freshwater ponds and streams, living under rocks. He or she, actually, the Planaria has, these are eye spots. They look like eyes, but they're not really eyes. They detect light and dark. And of course, if you're a little worm and every fish in the pond wants to eat you, you're going to seek out the dark spots under rocks and things. But they do have a very primitive brain. They have a gastrovascular cavity that runs down the middle. They have a nerve cord that runs on either side, and they have very primitive kidneys. So the protonephridia here are kidneys. And then they have one opening, which is their mouth, and the pharynx here. So they're pretty simple creatures. However, and this is a video of a Planaria that's been cut into 4 pieces. You can cut these guys into, I think it was 279 pieces, and get 279 identical flatworms. And this is actually an activity that I've done with a bunch of high school kids in the past. I mean, these things are amazing because within 24 hours, you start to see these pieces regenerate. And if you look at the white, right? So this was one of those middle pieces that's wriggling away because it's got white here, these are blastemas. This is where the regeneration is starting to occur. And you can see it's even got little tiny eye spots there on the end. And then as they continue to regenerate, you can see the blastema here and here. And within about 14 days, they've completely regenerated. And that leads to the question, how do they do this? How can you regenerate such an enormous part of your body in such a little time? And the truth to this is that these guys are filled with stem cells. So all these little green dots here are stem cells, absolutely filled. The only place they don't have stem cells is here. This is their feeding tube, and then right above the eye spots. So if you were to cut this off here, you would get regeneration of the eye spots. Well, the little piece with the eye spots would not form a whole Planaria, but the rest of the Planaria would regenerate its eye spots. And you can see that this group looked at the different types of germ cells, the different ectoderm, mesoderm, and endoderm, and showed that it's just filled with stem cells for the whole creature. There's a larger picture. So this is the paper. This is the paper that published this back in 2018. And the Stowers Institute is really leaders in Planaria research. And so to review this section on cells, it's important to remember that stem cells undergo asymmetric replication. They can repopulate the stem cell pool, and they show potency. There's 4 types of stem cells: totipotent, pluripotent, multipotent, and unipotent. And they're autologous, sorry about that, versus allogeneic sources of cells. Not all stem cells are the same. And, I'll leave you with why are Planaria an excellent model for stem cells and regeneration research in general. We're going to talk about cell signaling. So this is that third aspect of the circle that I showed you at the beginning, where we talked about cells, scaffolds, and now cell signaling. So when I was a kid in high school, this is how we used to talk to our neighbors when we weren't supposed to. We would pass notes back and forth. These days, people seem to talk to each other through their phones and their electronic devices. So cells do the same thing. They pass messages to one another. They talk to each other. They do not exist in a vacuum. So when we take, say, stem cells out of the tissue, skeletal muscle satellite cells, and grow them on plastic, it does not take them very long to lose their stem cell-like properties because they no longer have their neighbors to talk to. An example of this is cells exist in a tissue. So these will be muscle cells in a dish, which then form the lined myofibers in a tissue, which then come together to make up the skeletal muscle system, which then come together and make an organism such as, Arnold Schwarzenegger. So growth factors are part of the bioactive or one type of bioactive molecules that cells use to talk to each other. And there's different ways they can do this. A cell can talk to itself. That's called autocrine signaling. A cell can talk to a neighbor through these junctions that hold the cells together, or it can talk to a neighbor that's a little bit away. This is paracrine signaling. And then you can have a cell, say, in your muscle, talk to a cell in your kidney. And they do this through an endocrine system, through signals that are sent through the bloodstream. So there's multiple ways that cells talk to each other. And this is required for proper function of the tissue and the organism. And so very briefly, signal transduction refers to the conversion of signals from one form outside the cell to a response inside the cell. And that leads us back to our planaria. Why two heads and then heads or tails? So if you look at the planaria, this is our normal guy. And look at this one. This one has two heads. So why is that? Heads or tails? Well, the reason for that is that planaria and all tissues and organisms have polarity, meaning they have signals that say, this is my head and this is my tail. And in planaria, this is usually regulated by Wnt, the WNT, and then the NDK pathway. And so where NDK is high, you get eye spots. Where Wnt is high, you get a tail. Now, if you knock out one of these proteins, so if you knock out, say, the Wnt protein, so it doesn't know that there should be a tail. So you get two heads. Likewise, you can really mess them up by looking at APC or beta-catenin. And here you've got this poor planaria, has multiple tails, and this one has no heads. So the cell signaling is vital in embryogenesis, in forming the right, in signaling the right design and form of the tissue, but it's also very important in regeneration and letting the tissue know what it's supposed to be. So cell signaling, cells don't exist in a vacuum. They secrete factors that bind to cell membrane-bound receptors. Cell signaling allows for integrated tissue function, and this is really important when we talk about tissue engineering. And so, we'll get to this question in a little bit, but why is this an important consideration in regenerative medicine? So where are we in tissue engineering today? What is the state of the art? Well, if we consider the increasing engineered complexity of tissues, so this is flat, right? Flat tissues. Skin would be here, cornea, through tube-like blood vessels to hollow structures like bladder, and then to solid organs. We're pretty much here. We're still at the flat tubular type structures. Although there have been engineered bladders, we're still many years away from solid organs. So in practice, there is quite a few technologies that are being put into patients. This is an engineered trachea. This is the bladder that came out of that Dr. Atala and WFIRM have been putting into patients, heart valves, and then skin. So these are the really four big areas that are currently in clinical practice. And as I said, we're going to talk about some of the barriers. One of those barriers is our inability to expand cells in vitro or finding suitable alternatives to stem cells. And so remember, I said that, you know, I like to use skeletal muscle because that's my area of expertise, but skeletal muscle has satellite cells, which are resident stem cells. You need to be able to isolate those stem cells if you want to use cell therapy or make engineered muscle. The problem is the only cells that are available are generally from cadavers or discarded operating room tissue. And as you can imagine, neither one of them are great sources of cells, right? The cells are already pretty old, or they're being discarded because they're not functional. Inadequate vascularity. This is a big problem, and this actually goes back to when we were talking about scaffolds and how using a decellularized tissue, like the kidney or liver that I showed you, actually maintains the vasculature within the structure of the tissue architecture, and then scientific and technical scale-ups. Or this actually gets into biomanufacturing. And so we talk about scale-up. This is really the problem, right? So this picture on the left, this is from an experiment that I did about 10 years ago. This is a scaffold that I grew skeletal muscle cells on, and then I put them into the skin of a mouse to see if it would become vascularized and actually form muscle tissue. Now this scaffold is about 1.5 centimeters in size, and it's only a couple of millimeters thick, and there was skeletal muscle tissue on it. However, this is what we need, right? This is the skeletal muscle tissue that we need. And so how do we scale from this to this? And it's a problem. We haven't quite gotten there yet. And that also leads to the vasculature. The reason we haven't been able to make very large tissues or solid organs like kidney is because of this perfusion challenge. So oxygen can only perfuse about 3 millimeters into a tissue. Oxygen is required to keep the cells viable, to keep them alive. And if you can't deliver oxygen, then the cells die and become necrotic. So anything beyond 3 millimeters in size is going to require a vasculature. And I'm a very visual person, so these little gold cubes down here in the palm, these are about 3 millimeter cubed. So that's not a very big tissue size. This is a spinach leaf. But if you look at the spinach leaf, so let's go back to this. Oh, no, wrong way. These are the blood vessels in the leg, and you can see how they branch and they become smaller and smaller. Similarly, these are veins in a leaf, right? This is a spinach leaf. And there was a group about 4 years ago that showed that they could decellularize a spinach leaf. Similar to what we do with the heart, which you can do with the heart or the kidney or the liver. So you take all the cells out of it. And then what this group did, after they decellularized it, they checked the patency of the vasculature with a red dye. And that's what this video is. So they're injecting a red dye, and you can see that the blood vessels are still intact. Well, the vessels are still intact, right? They're still patent, they're still open. The next thing this group did was they placed cardiac myocytes. So these are heart stem cells. These are heart cells that normally don't grow very well outside of the heart. They injected the spinach leaves with heart stem cells, and what you see is that these stem cells contract. They spontaneously contract, which is what heart cells, that's what they're supposed to do. That's what myocytes are supposed to do. So the idea behind this, right, is that if you had multiple spinach leaves that you put cells on, and then you stack them one on top of the other, you could design a vascularized cardiac patch for a heart. And that's what this group was actually pushing to try to do. Develop a patch for damaged hearts. The last area that I want to talk about are some of the enabling technologies. And these are the technologies that are being developed that are allowing to push this, this field of science even further. One of the first things I want to talk about is mechanical preconditioning. And these are the bioreactors that we use. So when we talk about skeletal muscle again, during embryogenesis, right, a baby is constantly moving. So those muscles are constantly being stretched and pulled and moved around. And the muscle bioreactors, which you see here, do the exact same thing. It's a way to mature the tissue prior to putting it into an animal or a person. And you can see that it's stretching the muscle tissue very slowly, but continuously. It helps the cells align, helps them fuse to form fibers and mature. Nope, don't do it. Oh, the other blood vessel, or the other bioreactor we use quite often is the blood vessel bioreactor. So blood vessels are made up of two types of cells. Endothelial cells lie in the middle, the inside of the blood vessel, and smooth muscle cells line the outside. We have shown in the past that if you line the blood vessels with endothelial cells and then put it into an animal, once the blood goes through, that shear stress of the flow of blood actually will remove the endothelial cells. And so in order to maintain the endothelial structures inside the blood vessel, what we do is we put these into a bioreactor, and we can very slowly increase the shear flow of the media through that construct. And what that does is it teaches the endothelial cells how to stay in place for when you put that into an animal. So once again, we're maturing the tissue prior to putting into animals. Once again, if we were face to face, I'd ask you what this is. This is an inkjet printer, but it's actually one of the very first 3D bioprinters that was used at WFIRM. And so the way this worked was the ink cartridges that you buy, right, that we have to replace, it seems like every 3 months, the RGB, the colors, the black, the cyan, all those cartridges, they took the ink out and then put in either extracellular matrix like cartilage or cells, and then printed it on a flat sheet, just like an inkjet printer does. Today, this is our bioprinter. It's a little fancier, it's bigger, but it does essentially the same thing. It just allows a lot more control over the size and shape of what we're printing. And now it doesn't have to be flat. We can print 3D structures. Now, where this is going is to something like this. And so this is a design that we've actually been working on at WFIRM, and it was designed for burns. So you can imagine if a person has an abdominal burn, what you do is you put them under this printer. The printer first takes a scan of the patient, determines what layers are needed to cover that burn, and then goes back and covers it layer by layer, fully treating that burn. This is the 3D printing process. So really the first thing you have to do is scan the patient. So let's say we're trying to make a heart, right? You'd scan the patient's heart. You would develop a computerized 3-dimensional image of the heart. You would then make a model. You would have to put together Bioink, which I'll talk about in a minute. You would then print that, and then you would put it back into the patient, its application. So it's actually pretty simple, the whole 3D bioprinting process, the idea behind it. Bioinks are becoming very important. So you can't just print cells. The cells have to be in something to preserve their viability, as well as to provide structure to whatever it is you're trying to print. And in these different, this is what we call bioink. This is made up of polymers, cells, usually mixed together, so that when you do print your tissues or your constructs, you get the right form. And then these are some of the applications of 3D bioprinting. Skin and cartilage, some of the hollow tubes, tracheal splints, aortic valves, and then we've been working with the kidney, and most everybody has seen Dr. Atala's TED Talk, but if not, you can find it. It's pretty interesting on bioprinting kidney. One of the last things I want to talk about, one of the last enabling technologies, is this ex vivo console of human organoids, or the ECHO project. This is basically the body on a chip platform. You may or may not have heard of this, but it's also called organoids, an organoid platform. Basically, what an organoid is a very small piece of functional tissue. So it's made up of all of the cells that the tissue requires to be functional, and then put into a microfluidic chip. And so this is a microscope slide, and this slide actually has 4 wells for either 4 different organoids or 4 organoids of the same tissue. This is what the organoids look like under a microscope. So these are just different cell types stained with different markers, but you can see they're small, round pieces of tissue. And the important part is they're functional. Now, the nice idea behind this is that you can use this as a tool for drug screening using human tissue or human samples. And we're using this quite a bit to look at chemotherapeutic drugs on human tumor samples. But basically, the idea is you can put all of these different organoids, in series or parallel, and then treat them. So you can put a hormone in there. You could put a drug in there. You can run it through the lung first, or you can run it through the liver, since a lot of drugs are processed first in the liver. So you can add the drug here and then follow its effect on all these downstream, all the other downstream tissues. It's actually a very powerful platform. The reason this was developed was because there are a lot of deficiencies in the models that we've used before. So this is a mouse. Mice are not humans. We like to use mice a lot as an animal model because they're cheap and we can do very large experiments in short periods of time. However, mice, rats, they really don't mimic human biology. And there's a lot of ethical questions that are associated with the use of animals as well. The other model that we've relied on in the past is this 2D cell culture, where we take the cells out of the tissue, we grow them on plastic, right? The problem with that is that doesn't even remotely look like the organizational structure of the tissue that it came out of. And so these cells change very quickly over time when they're grown in this manner. So we wanted to develop a platform that will allow us to look at cells in 3 dimension that acted like, that had the physiological function of the whole organ. Part of this was including the microenvironment of the tissue. So the microenvironment is the extracellular matrix, everything that surrounds the cells and keeps the cells together. This is done in different ways. You can add extracellular matrix to your cells, or you can add fibroblasts or myofibroblasts. These are cells that automatically secrete the matrix, and they will secrete it for the tissue, for the organoid. At WFIRM, we've been working on this for quite a while, and this is just a list of all of our characterized and integrated organoids that we've worked on. These are the ones that we're working on now. The top ones in the light gray have been published. And that's where I'm going to end.
Yeah, Dr. Criswell, that was a fabulous lecture. I've got to tell you, as a biologist and somebody who really was very appreciative of cell biology, I thoroughly enjoyed that and feel like I got a tune-up. So I know that the people that are taking our beta skills, biomedical technology application skills, are going to really benefit by having taken this lecture today. So I want to thank you. And